Hello. Good morning, everyone. I'm very excited to present this keynote, and my very sincere apologies that I couldn't make it in person due to some last-minute, non-deterministic logistical reasons for my visa application. But anyway, here we are. So let's get started. Um, So today I'm going to talk about the landscape of genomics for system research, a topic quite close to my heart, and we have been working on it here at Michigan for the last 5 or 6 years. So let's start with this little, the trends in genome sequencing and why this is an exciting time to do this kind of research. So the very first human genome project was around, it created the first draft human genome somewhere around 2001. And it's amazing. I can't believe that it's just been about 22 years. And what we have seen is exponential growth in the number of genomes which are getting sequenced. So the very first genome which got sequenced in around 2001 was almost $300 million. It was a massive effort to do that. And since then, we have made a lot of progress. What we saw, the first was Sanger sequencing technologies and so on. And then we saw the next generation sequencing technologies come through. And the primary difference between these and a classic type of synthesis here is sequencing by synthesis. And what we saw the second generation, the big difference was high throughput parallel sequencing. There were these very efficient clustering methods and sequencing methods in parallel flow cells. And so we saw a bunch of Illumina machines around that time, and this was roughly a decade or more ago, right? Um, from second generation sequencing, we saw a growth spurt here, and we went to the thousand, we went to the thousand, we went to the 1,000 genome mark. And what we witnessed after that was the incumbent third generation sequencing technologies like PacBio machines and the more recent nanopore machines. And I'll talk a little bit about this this technology as well, but it dramatically reduced the cost, which means that we are going to see even a higher growth in the number of genomes which are being sequenced. And recently, the single sequence sequencing has taken on, it's become practical. And here we can do high resolution sequencing on a single cell basis, right? So imagine like before we could create one genome per human, and now with single cell sequencing, we can go and create so many different specific sequences for a single person based on which part of which cell and which organ and which tissue we are analyzing, right? So that again explodes the amount of sequencing which we need to do. At the same time, as the sequencing growth, number of genomes sequenced have been growing, right? The cost of sequencing has been going down. As I mentioned before, the first sequence ever done, the whole one, was $300 million, and or even higher to put it together. But that was around 2001 or so. And now what we see is that we can do a sequence, for almost $1,000 or even cheaper, depending on, the resolution and what you want to accomplish. So we see a dramatic fall in the sequencing cost. However, the Moore's law has not kept up with it. So now to meet that demand of sequencing, we need to have better systems, better hardware solutions, better software solutions to meet that genome sequencing demand, right? Because if the cost goes down, you're going to even sequence more. And in 2025, it's a target for many countries, right, including in Europe, to sequence all the citizens by 2025. So we can see a very high demand here. The let's take a look at the evolution of sequencing technology, and then we'll talk about the systems challenges here. So the Illumina sequencing by synthesis was the second generation technology. And I think what it reached was really the ability to sequence a whole genome in, whole genomes at the same time, right? So it could do 1 gigabases per day. And this is a 2021 machine, which reached 3 orders of magnitude higher. Base is like a single base in a DNA strand. And also these accuracy was very good, but however, when we used to sequence this, the fragments of DNA, which is called a read, which was created, was very short. So short reads gives you a smaller scope into the DNA, so which is very good for small variants calling, but then you cannot like use it for it's more challenging to use it for larger variants or even for assembling the genomes. So this is where we were. And what we see is a very exciting new technology which enables real-time sequencing, right? So this is a little like a biopore, like almost through which you can tug in the DNA strands and then measure the changes in voltage signal to figure out what base pairs are there in the DNA, right? So this was very interesting because it could really increase the length of the DNA strands which we could sequence at the same time. From 100 to 350 base pairs, we can go to kilobases, million, or even larger. However, the accuracy was a little bit lower here. So, let's going into the sequencing technology more, if you open this nanopore device, inside it, you will have lots and lots of flow cells, where each flow cell in a flow cell, you'll have lots and lots of pores, right? That's why you can parallelly do thousands of these DNA strands together. And each pore is just like a little motor protein, which helps you to attach the DNA strand, and a tiny biopore, which they call a nanopore, which tugs along the DNA string to itself, right? So if you can see, this is a very small device. So before, the sequencers were like size of a washing machine or a refrigerator. This is handheld. I can hold it in my palm, right? So that makes it very portable. And the other cool part about it is that you can carry it anywhere, and you can also do it real time. So there's a feedback mechanism which you can leverage when you build the system. So, it can do very long reads, greater than 4 million base pairs. It has simple library preparation. One thing which we found very interesting was also that it can do both RNA and DNA sequencing, which opened up the options for pathogen and microbiome application scenarios, which I'll talk about today too. And the low cost was very attractive, right? Like at $1,000, we could buy and set it up in our lab. The streaming output enabled real-time processing, which enabled many clinical use case scenarios, which were very exciting. One of which I'll talk about today. So what these progression of sequencing technology has led to is exploding sequencing applications, right? What we see is even in pandemic, and I'll show you how we could use sequencing technologies for early detection of pandemics, right? Or see the progression of how a pandemic progresses. And there was a lot of hype around consumer genotyping, which could tell you your ancestry and also your disease risks. One thing which I'm very excited about is how sequencing can help with cancer treatment, how it can do both diagnosis and measure the progression of treatment. For microbiome analysis, there's a lot of microbiomes in our body, and it can analyze that to give us a very good indication of our health. And it just has a lot of applications in agricultural sequencing. And with the move towards portable sequencers, we can even do sequencing in an operating room. when a patient is actually getting operated, if even in that short time frame, if we can sequence, I can do the margin detection, then that's a very exciting application. And today I'll talk about how we built some early results in that case and made that possible. And food safety is a very important phenomenon. The food industry, the fast food places, how they have to do the supply chain, manage their meat and poultry and so on, and it can get damaged. So and that can cause a lot of regulation problems, a lot of loss, wastage. And if we could have a pipeline with sequencing which can detect this early, that would be a fantastic application. And finally, liquid biopsy is very exciting. Here, if we know ahead of time, if we are going to get cancer, then there's a lot we can do to prevent it, right? And imagine like a blood test, which we do every year to do our markers, our lipid profiles and so on. If we could do that and we could detect these, detect if there is an onset of any kind of cancer, that would be fantastic, right? And there the computation problem is very significant because it's a needle in a haystack problem to find this free floating around cancer cells. And I'll talk about the portable pathogen detector, which is very relevant to today. We can use it for customer use cases. If we have any kind of pathogens and we're falling sick, how can we do it at the point of care and the point of use? So exciting set of applications, but these are all, not to make them work. To make them happen, we need a lot of systems research to make it work. So let me give an overview of how these applications can be actually placed in the broader context and what is a typical pipeline of almost all of these applications. Okay, so what I would call this is like a precision health platform. Now, the precision health platform would have many data sources. And these data sources are both digital and analog. So there's electronic health records, which records the patient history. And then we have biological signals like the genome, the epigenome. The epigenome is what it transcribes and forms. The microbiome, we would be surprised about how much microorganisms we have in our body. They outnumber human cells from 10:1 ratio. And then there's metabolome. These are small cells, like a complete set of small molecule chemicals that we find, like sugars or amino acids. So there are a lot of biological samples. There's our digital history, right? And then what is important is to convert these biological signals through some kind of mechanisms like sequencing to digitize that information. And electronic health records are, stored in a data store, right? So once you digitize this biological information, you can run data analytics on it, data analytics like statistics or neural network methods, and then you can create the diagnosis and prevention suggestions, right? Or treatment and management. How do we get from how do we manage and how do we kind of progress, build a health history? So that's the clinical decision side. So we start with data sources, both digital and biological. Sequencing helps to convert it into everything, digitize everything. And then we have analysis pipelines, which consist of both advanced statistical neural network methods as well as a process to kind of digitize the biological signals. So this is a typical precision health platform. And from systems design considerations, I feel there are three important challenges here. First is efficiency. Now, as I showed you, that the cost is dropping very fast and clearly outpacing Moore's law. So we really need very efficient solution. And just to give you a context, right? Let's say for Facebook user data, per user, you have few gigabytes of data. But with sequencing, you have terabytes of data. So there's a 3 orders of magnitude higher data processing needs here, right? And then security and privacy. This is our private information. So how do we make sure that, we can efficiently do this analysis and aggregate information across several users or patients to make meaningful insights and clinical decisions? and do that while preserving the privacy. So one example there could be using this GX technologies or homomorphic encryption. And form factor. The more we shrink it, the more we reduce the size and the weight of these devices, then we can push it to the edge, push it to the clinical solution, push it to doctors, to patients. Yeah. So there was this vision by, what if I sneeze into my device and it could just real-time sequence and tell me all the bugs I have? That would be fantastic, right? I don't have to go to I could figure out myself what's going on in my body. So these are some exciting challenges and systems considerations for building these platforms, right? So structure to it, and then what are the challenges there? Now, given that mindset, I would try to go into a few, genome sequencing applications, which we have worked on in our group and in Michigan in general, and show you how that panned out, right? And give some pointers as to future, what the future could be like. So one of the first application scenarios which we targeted was whole genome sequencing. This was back in 2017 or 18. Yeah, we published the first paper in 2018. And so this is how we got into the space. So to give you an idea of what whole genome sequencing entitles, right? So you have got the 6 billion, so you have DNA strands are roughly, these are these double helical structures. There's 3 billion pairs on each side. Together, there's 6 billion bases. Now, you get a sample, and how can we know exactly what this DNA sequence is? How do we digitize it? So the process goes like this. You take the sample, prepare it, and then send it through a sequencing instrument, right? Where it spits out lots of small fragments of the DNA as reads, right? And these reads have got these base pairs like A, T, C, G, right? So you have taken that long helical structure, got lots of little reads. Then it's really about solving this jigsaw puzzle. So you have these lots of small fragment reads, and you want to know where they fit in a reference genome. So we know we have a reference genome which represents, the common DNA DNA composition of humans, and we want to see where these individual patients' reads are kind of matching in that space. And after we do this alignment process where we have figured out the proximate location in the reference genome, we have to remove some data cleanup, right? So we do sorting and mark duplicates. That's not very intensive, but still an important process. And finally, the most important step, which is also the final step here, is to find out the variants. And this is the main goal of this process. We want to know how this patient differs from the reference genome, and where the differences are would have a clinical impact, right? Are they developing a tumor? Are they having some kind of indication of future critical diseases and so on? So that here, the reference genome pair is here. And what we see by doing statistical analysis, we can see that at this position, the C has flipped to an A. So that is the variant we are looking for. This is a single substitution variant. And finally, this, after we call the variant, we go to the diagnosis, right? So that's our pipeline. Now, if we analyze this pipeline, and this is a little bit, on one of the older AWS servers, but the rough proportions remain same. We see that if we break it down, a lot of time goes in alignment, and then we have sorting and mark duplicates, which is also like not a very, interesting to accelerate step, but takes up quite a bit of time. And then we have got this pair HMM, which is kind of inside the variant calling, which takes a lot of time, right? So we, in our group, we thought, okay, let's go for, accelerating this whole pipeline, all of these, right? And I'll show you solutions of all of these techniques and how they worked out. So, so let's see how we made progress. The first accelerator we proposed was GenX. Now, if you look at this alignment problem, the initial part is just an approximate string matching, right? Some things which computer scientists love to do. So, and a typical way of finding these approximate matching, like where a character was deleted or substituted, is using a Bandit-Smith-Waterman solution. Now that's great, but it's order of kn. In our group, we took a slightly different approach. We said, how about using automata? We are familiar with finite state machines. So how about we take an automata to solve this problem? And we built something called string independent Levenshtein automata. So there is already existing Levenshtein automata which can help you figure out the edit distances between two strings. But the problem with these canonical automata is that they're string dependent. So imagine if you have 3 billion base pairs and we have got, millions of these reads. For every little portion of the seek reference, there'll be a new independent automata. So we have to program millions of these automata and then do string processing on them. That was not scalable or feasible, right? Very soon we lost, we figured out that doesn't work. So what we did was, to make it string independent, we created a retro comparison code which would initially pre-process these strings and record these differences, right? And then we would pass this codes into a string independent automata, which would then do the processing. We also had a feature for in place traceback, which is which is less important now because traceback is very useful and very interesting. But what we figured out is that we can find the optimal read and only do it for that optimal location and only do traceback for that instead of for every single read alignment. And then we also had affine gap scoring, which is biologically relevant gap scoring, which is very important. Without this, if you just use edit distances, it doesn't give meaningful results. And finally, it had composability. How do we put these little machines together? We built a ASIC chip for it, and it was very fast for read alignment over a canonical solution, a CPU or GPU. We further refined this to figure out if we can improve the utilization of our processing elements. So here is a problem, right? So this was our original solution where we would go with a wide band, like this is the diagonal wavefront on which the P's are computing their optimal scores. What we found is most of the accurate paths are somewhere in this region, and the rest of it has low utilization. So to provision so much P's to process all these cells in our dynamic programming matrix was inefficient. So instead, we could look at a banded implementation, which will have a narrow PE array and only analyze a small portion of the solution, the green portion, and will not do the rest. But how do we ensure that we do not miss an optimal path? For that, what we did was we figured out a carefully curated set of optimality checks. We figured out the scoring threshold at these boundaries, like what's the max possible best score which you can get there, right? And if the score which you're getting here is higher than that upper bound score, right? It's higher than the threshold. There's no point of evaluating this region of the dynamic programming matrix. So we can skip it. There was a little problem here because it's an asymmetric matrix, because usually reference strings are larger than the query strings. And for this little asymmetric portion, we figured out an edit distance score, which is even more optimistic than the threshold-based scheme, but it used to filter out a lot of cases. That ways, we could do a very efficient, area-efficient alignment algorithm. And this paper we published in Micro recently. In 2021, GenAx was published before that in ISCA. So we managed to make sure, and we paid a lot of attention to accuracy here and equivalent result. It was 100% equivalent, and it was built on FPGA F1 Cloud on AWS. So we actually went and made, not just designed it, but created a verified FPGA implementation for this, which was integrated with BWMM software. And it gave us a 6x throughput benefit. Finally, we looked at also not just the seeding, but also the alignment problem. And this was a very interesting scenario, because what we saw is that to figure out the candidate locations on the reference, like you have this read and you want to figure out these candidate locations, right? And there's several of these candidate locations. What we found is that the most popular data structure which is used to do this, called FM index, is actually using a lot of complex representations, which lead to a lot of memory bandwidth bottleneck. So it's very small, right? It's few gigabytes big, this index, FM index, right? So here is the size, right? So it's about few gigabytes, but it requires a lot of memory bandwidth and a lot of data to be fetched uh, to the CPU. And because of that, it was getting very slow. So our insight was, can we create a data structure, which is a hardware software co-design? It increases the memory capacity, right? So we saw that the memory capacity was increasing, but it had low data requirements. It didn't fetch that much of data per read. And because of that, because of the lower memory bandwidth requirement, now we could really speed up. That bottleneck was gone, right? So this is a true hardware software co-design project where we as architects could understand what the bottlenecks are and redesign this data structure. Now, the bioscientists thought, okay, the more we compress it, the more small we make this index, that's good, right? To fit it in the computer memory. But that's not the case. We have now 128 gigabyte RAM, so 512 gigabytes RAM. With memory expansion, we can go to terabytes too. So this is something we did. And then the data structure which we proposed was called enumerated radix trees, where we had a lot of clever optimizations, but the key idea was to build a radix tree and have special sort of nodes to capture locality or long strings. The other very interesting thing we did was, when we're doing these searches, right, over the indexes, we skipped a lot of searches by looking at some bandwidth efficient search algorithms there. So that was our work, which was published in ISCA a couple of years ago, or a year ago, I forget now. But yeah, so this is ERT. So one thing which was useful here is that we proposed a hardware accelerator, but this data structure which we proposed could just speed up the software. And so we went to BWMM folks, and BWMM is the de facto genomics read alignment tool. And they've improved it with Intel's collaboration to BWMM2 recently. And we went to those folks and said, hey, do you think this data structure is useful? And we actually got that integrated into the GitHub and is being used by quite a few people around the world. So not only hardware innovations, you can push software innovations, right? Finally, we also worked on this sorting and duplicating pipeline and created, we saw that this was IO bandwidth bound, and we figured out some sorting implementations and a main memory implementation which would not go to the disk so much. And that helped to reduce it quite a bit, right? So the final step was variant calling. As I was telling you, this is about the differences. And here also we adopted, like CDEX, a pruning mechanism. What we found is that when we are doing this variant calling, again, it's a dynamic programming problem. And lot of the cells in this dynamic problem solutions are not necessary for accuracy. Only few cells are very important for accuracy, and we can prune away the rest or do it in lower precision. So what we ended up doing was, floating point PE for these critical ones, and then approximate log-based PE to figure out the pruning, like which cells are important, right? Which cells dominate the computation. So that was our accelerator architecture. We also taped out a chip for it. And it gave us, it was bit equivalent, right? Like all our work, we focused on bit equivalency here for high accuracy. And it was 43x fewer cells and gave us an order of magnitude speed up. Now, throughout this whole pipeline process, if you would notice, I have paid a lot of attention on accuracy. Why? Because this accuracy matter. And this is a slide from a collaborator and a friend, Onur Mutlu. But, I really like this slide, right? So this kind of tells you why accuracy is important, like, even within human, there's only 0.1% difference, right? And if you go below 0.1%, I don't know if you're identifying a cat or a chimpanzee or so on, right? So even very minuscule differences will make a huge difference to these biological pipelines, and they have been carefully curated and validated. So please pay attention to accuracy when you develop these genome sequencing pipelines and hardware accelerators. Here's a little demo from my group.
In this demo, we show how ERT and CDEX can be used to align reads about twice as fast as software VWMM2 and call variants in the CYP2C19 gene, widely responsible for drug metabolism. After downloading the alignment outputs, we now move to the location of the CYP2C19 gene in chromosome 10. Each red block corresponds to an aligned read. We can see that more than 50% of the reads have a G2A variant in this gene.
So here my student is showing how we identified this variant here, right? Now let me explain you how this variant actually makes a difference, right? So this is a drug metabolizing variant. So when you take a DNA, usually the DNA kind of guides the protein formation, right? And how it works is that certain markers help in transcription. So here, this marker, here what we are seeing is G2. So anything between GT and AG here is spliced, and the remaining part of the DNA, these are the non-coding regions, is kept, right? So the non-coding is intron is kept out. Now, if we make a mistake and flip this A to a G, if we flip this, what we see here is if we have made a flip, then suddenly we see an AG appear here, right, instead of here, and we cut out a long non-coding region. So instead of cutting out a small non-coding region, just because of one mistake, we are going to splice out a big region, and the transcription would be completely wrong, right? So we'll not get a normal RNA, but an aberrant RNA. And because of this aberrant RNA, the normal protein would be able to metabolize common drugs like blood thinners, but this aberrant RNA will not. So it's very important if a patient has this aberration that we should correctly identify it. We shouldn't make mistakes in the sequencing pipeline, right? So this was an example for that. Okay, so with all our acceleration study, we could get a 5.5x reduction with an FPG implementation. With an ASIC, it could have been more. But what we saw was that the non-accelerated portion was actually leading to a von Neumann bottleneck. And so there is opportunities to kind of further accelerate that part of the pipeline. But overall, this was an end-to-end pipeline, which was bit equivalent with the original software. Okay, so now I showed you how we took existing software, didn't really change the algorithms, right? Maybe we changed the data structures and then built really efficient systems. But what followed on was that with all our expertise and all the knowledge we gained here, a few faculty in my group actually went on to develop sequencing tools, which is being used, right? So this is going and developing brand new algorithms for bioinformaticians, right? So let's see what they did. So this is a recent work which was published in Nature Communications just last year. And what they observed is that the ways the variant caller represents is very dependent on alignment. Here, what it is showing is that the same reference and query, there are many different ways to align it, and the variant caller is not independent of these alignments. It gives a different score for all these possible alignments, right? This makes it very challenging to compare the accuracy of different variant calling tools. And this was a big pain point among in the sequencing community, right? So they went on and built this tool which would made it independent of these edits, right? And could give a very high accuracy. And now we have folks from PacBio, NIST, which is the National Institute of Standards and Technology, which develops all these standards, and people in Michigan messages reaching out to use this tool, right? So not only systems design, if you develop that expertise, you can go and build bioinformatics tool, which can be widely used. Now, I'll give you another example of where we went a mile ahead. So we started with hardware acceleration. We went to even tools development. And then in my group, we also looked at wet lab, build a wet lab. So why did we need to do that? So let's take a look at this. So this is a very interesting application. So for neurosurgery, right? So if you have a brain tumor, what they do is during the operation, they take a slice out of your brain tissue and then examine it under a very high resolution microscope, like a histology slide, and then tell you whether that tissue has aberrations or tumor cells or not. And that's how they detect the margin of the tumor. So And this margin detection is so important because you don't want to cut out, tissue unnecessarily from the brain, right? That can impair you. So this is a frozen section histology method, very outdated method. What has happened in the recent years is that now people have developed genetic markers for tumor cells. And so we can potentially use sequencing to do this margin detection. So, our partner physicians, neurosurgeons in the Michigan Medicine came to us and said, hey, we have a challenge for you. You're working on real-time sequencing, but can you really do a tumor DNA sequencing in intraoperative time frame? When the patient is open on the operating table, can you do the sequencing in less than an hour or in half an hour so that we can beat the histology results, right? Make it more accurate. So we took up on this challenge. We started building a whole pipeline, right? So from DNA extraction, you take a brain tissue, you extract the DNA, you do the amplification, library preparation. We do the sequencing on MinION, which was a real-time sequencer. We cannot do this on Illumina because that takes too much time and it is batched. Okay, so we had to take the real-time MinION sequence and then we do informatics. And what we saw when we're doing this is that, hey, the informatics part of it was really fast. In fact, the sequencing was fast too, right? It was really fast. So where was the time really going? And what we figured out is that the time is going in something which we have no control over. It's the amplification process, the PCR amplification process. Now we all know PCR because of COVID, right? It amplifies the DNA. So yeah, 1 second. My alarm has gone up. 1 second. Yes. So the whole thing, time was going in the wet lab process of target amplification. Now, what do we do about that? Should we give up? Well, no. What we did was we looked at solutions which can attack this. But for that, we had to set up a wet lab at Michigan. So what we did was, okay. What we did was we set up a wet lab at Michigan, and this is a postdoc, Jack, who had joined our lab at that time. It was a biosafety level wet lab where we could do actually sequencing and amplification and all that. And now he loves this stuff so much. He's in Michigan Medicine, actually. He graduated from our lab and went on to join the doctors. But yeah, so we set up this wet lab, and then we looked at some co-optimization of wet lab and hardware systems, right? So we looked at process of threshold sequencing, like how less can you sequence and do more in the informatics side. So that gave us some results, and it reduced it to about 52 minutes, right? Which was great. It was the first sub 1 hour sequencing demonstration. But the target application was still a bottleneck. So then we went and did something very crazy. We started looking at isothermal technique. So the main bottleneck with PCR was they have to do thermal cycling. So it's a process where you increase and reduce the temperature, very mechanical, and it's very hard to reduce the time there. But isothermal techniques, we do not do thermal cycling or temperature changes. And use chemicals to amplify, like loop-mediated isothermal LAMP technologies, are very fast, right? The main problem there is like they create a lot of bad product, lot of erroneous product, which we could informatically correct. So we could put our computer science hat on and informatically correct this. So we built this tool called Lamprey to work with LAMP amplification, and with that informatic correction and LAMP, we could get it down to, less than 30-minute sequencing threshold, and that set a record for fastest time to result. All right, so I'm rushing through this because we are kind of getting out of time, but I just wanted to give you a spirit of this research here. Finally, we also looked at real-time pathogen detection. Now, we have lived through the pandemics, and we're seeing rise of superbugs like bacteria, which are resistant to antibiotics. And so, bugs and pathogens are making a big, are a big and important part of the sequencing circle. And we thought, can we develop some pipelines for this? So for that, we look at COVID testing, right? So when COVID came, you take the swab and do amplification, and that's how you figure out the presence or absence of COVID. But to create this whole pipeline, right, like the PCR amplification, the liquids, and then to pass them around, that takes a lot of time, and it is also very error prone, as we could see, right? Initially, it took months for COVID testing capabilities. What if we could create digital sniffers based on the sequence, based on their RNA sequence? If you could make digital sniffers, distribute it all through the world just through some data transfer, and then everybody could sequence independently and figure out what is this novel pathogen which is emerging. That would drastically cut this time and create a universal pathogen detection, right? And by this collusion and analysis, we could figure out whether a new pandemic is evolving. So this is a very exciting application of sequencing. And the advantage is real-time, portable. You have digital primers. You don't have to make wet lab primers and distribute it all over the world physically, right? So you can just digitally distribute these primers, and you can detect unknown pathogens which you have not seen so far, using, and also do disease progression with stain typing. So we developed this pipeline where we would take specimen, do the extraction through the with thousands of cycles. We could conduct clinical trials, and we have a base caller, and then we can do an accelerated filter, which is where the systems stuff, and then we can do sequence analysis and interpretation with machine learning. So the solutions we developed was this interesting feature of MinION, which we used was read until. So here what we realized was that 99% of the DNA, which after reverse transcription, what we get is host or human DNA. Only 1% is pathogen. So we want to skip those DNA. And what we can do is, in the real time, as we're sequencing the DNA of the host, we can detect it, have some classifier, and early on, within just reading a small part of the read, we can figure out and eject it out of the pore, right? So that was the read until part of it. And then if we have enough confidence in our call, then we can stop the whole process. So these were the real-time approach for pathogen detection. And we also figured out a way to skip the base caller, which takes a lot of time. So we could analyze the raw voltage signals from an ion and do that. And that's what we called a squiggle filter, and which gave us a lot of efficiency improvements. Not only for virus detection, we also looked at the composition or pathogen abundance of bacteria. And this is results from our wet lab, which took us just $90 an hour. And we could see that this pie chart is what is actual proportion. And this is what our tool gave us after analysis, which is pretty close. So many different ways to do this. And how can you kickstart genomic research in your group? Well, we also have released a genomics benchmark. It's open source. It covers 12 computationally intensive workloads and both short and long read. So please go and take a look if you are interested in this. And how benchmarks lead to ideas? Well, this is from our recent ISCA paper. We're building this genomics benchmark, which covers 3 different pipelines, as I've explained in this talk. And what we found was a very common primitive here was dynamic programming. And we have built dynamic programming accelerators independently in our group. But won't it be nice if we could find one programmable DP accelerator? It is like a TPU, right? Like in matrix multiplication is the heart of machine learning, and a systolic array could solve it. Dynamic programming looks like the heart of genomics. So we wanted to build like a dynamic programming is a fundamental as an accelerator. We analyzed all the DP kernels, and although DP, the main computation primitive was dynamic programming, there are many differences, right? So the dimensions, like it was 2D or 1D, the dependency could be wavefront versus graph structure, the data type and precision needed was different. So based on all these differences, we had to make sure that it works for all these different variants of dynamic programming. So the divergence of dynamic programming made it challenging to design this accelerator, but similarity is where we could customize. And we developed this GenDP framework, and I won't have time to go into a lot of details here, but essentially it had 2 parts which looked at intra-cell, how can we do that more in a domain, in a broader domain-specific manner, as well as the dependencies, how can we handle that in the intercell analysis. So we had local dependency, a one-dimensional systolic array, and then we have reduction tree path, which captured the objective functions. And also we looked at the differences such as precisions and dependency patterns and customized for it. So you can have more information on this in ISCA paper. And this gave us a pretty large order of magnitude speed up. So I gave you a lot of flavor of all the research we have done, and this is done by a team of faculty at Michigan. It's definitely not myself. I've collaborated with Satish and David on this, and Jenna for machine learning, and our partners in Michigan Medicine, Bob Dixon, who is a pulmonary and critical care medicine, and Carl Koshman, who is a neuroscientist, neurologist. So that's our team, and of course none of this could have been done by an awesome group of fantastic students. Jack and Zhao were our postdocs, and others were our PhD students. Most of them have graduated except you, Feng, who's still around and who did the GenDP work. And that's it. That's my talk. Thank you very much for listening, and I'll take questions now.